Mycology Part 3, some more features of fungi. We saw 4 categories of fungi: yeast, yeast-like, filamentous and dimorphic. Let us have a look at some more features of these 4 categories. The yeast, they occur in the form of round or oval bodies which reproduce by the formation of buds known as blastospores. Yeast colonies, they resemble bacterial colonies in consistency and in appearance. And the only pathogenic yeast in medical mycology is CRYPTOCOCCUS neoformans. This is a photograph of yeast colony, that is colony of CRYPTOCOCCUS neoformans. It resembles bacterial colonies. CRYPTOCOCCUS neoformans has a big capsule around it, and this big capsule can be seen by some capsular stains, and in the microscopy, when this capsule is seen by fluorescent stain, you can see this first photograph which shows the fluorescent stain in which the capsule is seen around the yeast cell, that is CRYPTOCOCCUS neoformans surrounded by a fluorescent capsule. In the second photograph, it shows a big capsule surrounding the yeast CRYPTOCOCCUS neoformans in the Indian ink preparation. And the third photograph is just a electron microscopic photograph. Yeast-like, that is the second category. These are fungi which occur in the form of budding yeast-like cells and as chains of elongated unbranched filamentous cells which present the appearance of broad septate hyphae. So, sequence of cells, elongated cells arranged in chain, it looks as if it is a hypha, but it is not a hypha, so it is known as pseudohypha, and a mass of such pseudohyphae is known as a pseudomycelium. The yeast-like fungi are grouped together in the genus Candida. So, example of yeast-like fungi include Candida. Colonies of Candida, they also resemble colonies of bacteria. They are also smooth in consistency and they resemble bacterial colonies. Pseudohyphae and pseudomycelium means hyphae जैसा लगता है लेकिन वो hyphae नहीं है. It is just a chain of elongated cells. It looks like hyphae but it is not a hypha, so it is known as pseudohypha, and a mass of pseudohypha is known as pseudomycelium. Candida albicans में germ tube करके एक property होती है. Germ tube is an outgrowth produced by the spores of spore releasing fungi during germination. The germ tube differentiates and grows and develops by mitosis to create somatic hyphae. A germ tube test is a diagnostic test in which sample of fungal spores are suspended in serum and examined by microscopy for the detection of any germ tubes. So, germ tube is a protrusion coming out of the yeast cell. It is particularly indicated for colonies of white or cream color on fungal culture, where a positive germ tube test is strongly indicative of Candida albicans. So, germ tube test positive is a property of Candida albicans. Here you can see in the photograph a tube-like structure arising from the yeast cell. This tube-like structure arising from the yeast cell is known as a germ tube. The first photograph is of lactophenol cotton blue mound LCB preparation. The second photograph is gram stain, and the third photograph is from electron microscope. All these photographs, they show tube-like structure arising from the yeast cell, which is known as germ tube. It is a property of Candida albicans. The third category, filamentous fungi or MOULDS. The filamentous fungi, they have a body known as thallus, and from the thallus hyphae they arise, and they are true hyphae, and the hyphae, they can be septate or aseptate, and at the end of the hyphae there can be some structures, specialized structures like phyalides and medullae, and at the end there can be some spores, the fungal spores. These are known as conidia. Here in this figure you can see this photograph of aspergillus, which shows lots of fungal spores at the end of the hyphae. A vesicle-like structure is formed, and at the end of the vesicle there are fungal spores. The fourth category, thermally dimorphic fungi. They are having two morphologies at two different temperatures. They are having yeast form at 37 degrees Celsius and mycelial form at 27 degrees Celsius. So this is called thermal dimorphism. Example, HISTOPLASMA capsulatum. Here you can see the colonies of HISTOPLASMA capsulatum at 27 degrees Celsius or environmental temperature. They are rough colonies because they are having filamentous form, that is environmental temperature, and at 37 degrees Celsius or human body temperature, they are showing yeast form or plain, oval or spherical yeast forms are seen. So at two different temperatures, two different morphologies. This is called thermal dimorphism. It is observed in dimorphic fungi. Then reproduction in fungi. Reproduction in fungi can be of two types, asexual or sexual. In asexual spores, there are several kinds of spores observed. Sporangiospores and conidiospores are examples of asexual spores. Sporangiospores means spores are formed in the sporangium, which is a bag-like structure. It is observed in zygomycetes, example Rhizopus and Mucor. And conidiospores, they are borne externally on the sides, example in Aspergillus, Penicillium and Dermatophytes. And conidia can be of two types. Small ones, they are known as microconidia, and the large ones, they are known as macroconidia. Macroconidia can be multiple celled or single celled, while the macroconidia are single celled spores. Sexual spores can be of four types: oospores, zygospores, ascospores and basidiospores. Then there are other vegetative spores: blastospores, a fungal spore that arises by budding; arthrospores, certain spores which they appear in the form of a string of beads. They are formed by fission. They are known as arthrospores. Then chlamydospores, a thick-walled intercalary or terminal asexual fungal spore formed by the rounding up of cell, and it is not shed. This is known as chlamydospore. Then phyalospores, a type of conidium found, for example, in many of the urotales and other fungi. Phyalospores develop at the tips of specialized finger-like cells termed as phyalides. So different kinds of fungal spores are observed. Here in this figure we can see sporangiospores in the first figure, and a sporangium is a bag-like structure in which sporangiospores are there, and the hyphal stick which supports this sporangium is known as sporangiophore, right? So sporangiophore has a sporangium which has sporangiospores in it. Then the second figure is of conidiospores. These conidiospores are spores borne externally on the sides. Then the third figure is figure of blastospores. Blastospores are spores formed by budding. Fourth is chlamydospores. Chlamydospores, rounding up of cells takes place, and double-walled rounding up of cells is seen at the terminal or intercalary junctions. These are known as chlamydospores. And the fifth one are arthrospores. They look like chain of beads. Chain of beads like appearance is seen in arthrospores. Fungal infections are of four types: superficial mycosis, cutaneous mycosis, subcutaneous mycosis and systemic mycosis. Superficial mycosis is surface infections limited to outermost layers of skin and hair. The cutaneous mycosis, fungal infections extending deeper into the epidermis and its integuments. Subcutaneous mycosis extending into the subcutaneous tissue, muscles and fascia. And systemic mycosis, infections originating primarily in the lungs and spreading to other organs. And there is one fifth category which is known as opportunistic mycosis. It can be of any of the above four categories. An infection occurring under immunocompromised state is known as opportunistic mycosis, and such fungi are known as opportunistic fungi. With this we end the part 3. In the part 4 we will see laboratory diagnosis of fungal infections. Thank you.